All right, everyone, we're back here for our part 2 of the chemistry lecture. We're going to pick up here with an overview of organic compounds. So our organic compounds are carbohydrates, lipids, proteins, and nucleic acids. Those are our macromolecules that we find in our bodies, and those are the major molecules that are going to come together to give us the cell. They're going to make up the cellular level of structural organization. So through this presentation, we're going to be talking about the structure of each of these different macromolecules, but also its importance within the body. So first and foremost, since these are organic compounds, we will know that organic compounds will always contain carbon. We went over this in our part 1 of the chemistry lecture. So organic compounds always contain carbon. Carbon is such an important molecule because it can form 4 different covalent bonds with other different atoms. And this gives us the ability to form very large molecules that can be combined into different shapes and so forth. So the carbon is a very important an important atom, and that's going to be integral to our understanding of organic compounds. Now, carbon compounds have a lot of very interesting characteristics. So first and foremost, they serve as a good source of energy. So carbohydrates are going to be one form of energy that we can find within our bodies. When we break down carbohydrates, there's a lot of energy that is stored in that, and that's actually how we're going to be creating energy in the body. Our carbon compounds do tend to be hydrophobic, which means that they don't like water. They do not easily dissolve in water. And our macromolecules will exist within the polymer form. So there's a few different terms that I want us to get into here so that we can understand organic compounds. And when I use this terminology, I want you guys to understand what I'm saying here. So a monomer is a single subunit of one of these different types of organic compounds. So lipids will have monomers, carbohydrates will have monomers, and so will proteins. We have different monomers of each of these different organic compounds. And what monomers are just a single part. So a polymer is going to be a chain of monomers. So these macromolecules are polymers of their monomer substituents. And so each of these different organic compounds is going to have their own unique monomers, and they're going to be in the structure of different polymers. So it's important to remember from our last video these two reactions here, the dehydration synthesis and the hydrolysis reactions. We can remember that we did this briefly in the last class. So the dehydration synthesis is going to remove water and create a bond, and you can see that we take these two individual molecules here, and we're going to combine them to get this and water. And so we can see over here that glucose and fructose are going to be combined to give us sucrose, and then we have some water over here also. So glucose and fructose are two different types of carbohydrates. And because they are two different types of carbohydrates, that makes them monomers. So glucose and fructose are two individual subunits of carbohydrates. And when we bring them together, we say that this is a very simple polymer, but this is a polymer because there's more than one. There's more than one carbohydrate here. So sucrose is just the name for a glucose and a fructose molecule that has been combined. So the dehydration synthesis reaction is going to be the reaction that's going to be building up these different molecules that we're going to be talking about. And then the hydrolysis reaction is going to be responsible for breaking those molecules down in catabolic reactions. So a monomer is a single subunit, and a polymer is a chain of those monomers. Each macromolecule or organic compound we're going to talk about is going to have their own unique monomers, and therefore that's why their polymeric structure is always going to be unique. So we're going to start here with carbohydrates. As I mentioned earlier, carbohydrates provide much of the energy that we use throughout our daily lives. And when we get to chapter 25, we're actually going to learn how carbohydrates can turn into energy within our bodies. Now, as I also just said, each one of these organic compounds is going to have their own monomer. So the monomer of carbohydrates is what is known as the monosaccharide. The monosaccharide, you might have heard of this word before. The monosaccharide refers to a single monomer of carbohydrates. So a single carbohydrate is going to be a monosaccharide. So this suffix here, saccharide, is going to indicate that we're talking about some type of carbohydrate, and mono means one. So this is one unit of a carbohydrate. So we have five different carbohydrates over here. We have deoxyribose and ribose. Those are our pentoses. And we have some hexoses, which are the glucose, fructose, and galactose. Now, pentoses refer to the amount of carbons that exist within the molecule. So I know this might be a little bit beyond where we're at, but what you can see here with these different structures is that there's this C here, and then there's this strange pentagon-looking shape here. Well, we're not going to get into the organic chemistry of why this molecule looks this way, but what I'll tell you is each one of these corners here, every place that there's a little corner, that stands for another carbon. So you can see that there's 5 carbons included in deoxyribose and 5 carbons included in ribose. The hexoses indicate that we have 6 carbons that can be found within our monosaccharides. So glucose, you can see we have one here, here, and here, and that's a total of 6 different carbons included in this molecule. So these are the 5 major monosaccharides that are going to be used to create carbohydrates as they can be found in our bodies. So again, on the last slide, we combined glucose and fructose to give us sucrose, and so that is an example of a dehydration synthesis reaction and the formation of a polymer. But maybe we have a molecule that's going to take a glucose and link it to a fructose, and then to a galactose, and then maybe to another fructose, and you can see that the polymer is going to be much longer and longer and longer. So one thing that I want to mention is that when we have two, in this picture where we had sucrose here, I mentioned that this was considered a polymer. Well, it is because there's more than one, but we would more accurately say that this is a disaccharide. So glucose is a monosaccharide, and fructose is a monosaccharide. And then when we combine the two of them, we call that a disaccharide. Disaccharide for 2 saccharides. If we were to have 3, we would call them trisaccharides, and so forth, and so forth. We also have oligosaccharides, which is sort of a bunch of different carbohydrates that have all been linked together. But the term that we will more often use to depict a long chain of carbohydrates is a polysaccharide. So polysaccharides are going to indicate that we have a very, very long chain of monosaccharides, or a long chain of the monomer form of carbohydrates. So you can see 2 examples of disaccharides here. You can see galactose and glucose have come together here to give us lactose, and 2 glucose molecules have come together to give us maltose. So each of these are considered disaccharides because there are 2 monosaccharides within each molecule. Now that these are linked, these are disaccharides. So as I also just mentioned, polysaccharides refer to a long chain of monomers. Remember, this suffix here, saccharide, refers to carbohydrates. So a polysaccharide is a long chain of carbohydrates. Now this carbohydrate, this polysaccharide here, is a very, very special carbohydrate. This is known as glycogen. Glycogen is going to be a very, very unique type of carbohydrate. But we also have cellulose in our bodies, and we have starches found in our bodies as well. But the one that we want to mention, these are all different versions of polysaccharide. They're all going to be unique in their own way. But the one that I want to mention is glycogen. Glycogen is so important because it is the primary storage polysaccharide in the human body. Now, when I say storage polysaccharide, what am I referring to? Well, when I say storage, when it's the primary storage polysaccharide in our bodies, it's going to be storing energy. And that's what it's going to be doing. We're going to be storing energy in this, and then we're going to be able to convert it later. So glycogen is going to be essentially, the structure of glycogen is a whole bunch of glucose monomers that have all been linked together. So you can see these little hexagons here, these purple hexagons. These are depicting the hexose glucose. Hexose, remember, that's going to have 6 carbons, and each one of these is going to be a glucose. So we have a glucose here and a glucose here and so forth. So all of these are glucose molecules. So that's what glycogen is. It's just a bunch of glucose molecules that have been together. There's no interesting pattern. There's no other monosaccharides that we need to worry about in glycogen. It's only glucose. And so glycogen is going to store this glucose, and then when our body needs to start breaking down carbohydrates for energy, we're going to start to liberate these glucose molecules from the overall polysaccharide structure. So this glycogen is going to be primarily stored within the liver. Now again, when we get into chapter 25, we're really going to talk a lot more about how glucose is broken down and turned into energy. But I want you for right now to note that glycogen is our primary storage polysaccharide in the body. This is going to be the major polysaccharide we're going to mention throughout this class. So I want you to make sure that you are comfortable with glycogen. So in review here, our monomer of carbohydrates is a monosaccharide, while the polymer is a polysaccharide. Remember, poly means more than one, so the polysaccharides are going to be more than one monomer linked together. Glucose is one of our hexose monosaccharides, and when we link a whole bunch of glucose molecules together, that's going to give us glycogen. And glycogen is going to be storing glucose so that we can use glucose later to create energy. So next up are the lipids. Now there are different types of lipids that can be found within the body, and we have different monomers for our lipids as well. So what we're going to start up here with is fatty acids. Fatty acids are long hydrocarbon tails, which are just made up of hydrogen and carbons, and they're these very, very long tails, and they can be used to create different things. So they can be used to create phospholipids, which we see down here. And we can use these to create triglycerides, which we can see here. So there's a few different things that we can do with our fatty acids. But as I mentioned, there are a few different types of lipids, and based on the type of lipid, that's going to give us our function. So of course, these will be different in their structure as well. Triglycerides are going to be made up of 3 fatty acids and a glycerol backbone. And I'll show you what a glycerol backbone looks like on the next page. But triglycerides are going to be the type of lipids that are stored in our adipocytes, also known as our fat cells. So the triglycerides are going to be important for protection as they're going to cushion different organs. The kidneys are going to be surrounded by a lot of fat, and that's going to protect the kidneys. This is going to insulate the body to keep heat within the body, and it's also going to function as an energy storage. And so we can actually break down fats in order to produce energy. If we run out of carbohydrates, we're going to be able to use fat. So we're going to talk about that in chapter 15. Phospholipids, on the other hand, is another major type of lipid that we see. Phospholipids are a very special type of lipid, and they are going to be found composing up our cell's membrane. So the cell membrane is going to be made up of lots of different proteins and such, but the major component are going to be these different phospholipids. And I will talk a lot more about phospholipids in later slides, and especially in chapter 3. But these are going to be the major types of lipids. So we have fatty acids that are going to help to create these different other types of lipids. Now on this picture, what we can see is that fatty acids are shown over here on the left side here. We can see these fatty acids right here. So a fatty acid is just a long hydrocarbon tail. You can see that we have a bunch of carbon molecules that are all bound together in the center, and then there's a bunch of hydrogens that can be found on the outsides. So this is a hydrocarbon tail, and there's some other molecules up here, but we will touch on these later. So one of the things that we can see with fatty acids is that fatty acids can be either saturated or unsaturated. And so again, you may have seen this whenever you've looked at the nutrition labels on your favorite food that you get from the grocery store. But when you look at these, saturated and unsaturated fatty acids are going to be looking a little different. So the only difference between a saturated and an unsaturated fatty acid is this little double bond right here. So you can see most of the bonds in each of these saturated and unsaturated fatty acids are going to be single bonds. There's just going to be a single covalent bond. But we have a double bond here, and based on the stereochemistry that's taking place at this double bond, that's going to cause this fatty acid tail to kink and move to the side. It's going to cause the bending here of this tail. And so these are the unsaturated fatty acids, and these are the saturated fatty acids. So unsaturated fatty acids tend to be liquids at room temperature. So things like your olive oil or your different types of oils that you'll find in the kitchen. Those are different unsaturated fatty acids. While something such as butter is going to contain lots of saturated fatty acids because butter is a solid at room temperature. Now, the importance of saturated versus unsaturated fatty acids. Saturated fatty acids all are going to be straight, like this fatty acid right here. So they're all going to be straight. Now, if we had a few other saturated fatty acids, these would be able to stack very nicely with one another. They'd be able to be very closely associated. And there are certain different interactions that are going to occur, and that makes it a lot harder for us to break the bonds between two saturated fatty acids compared to two unsaturated fatty acids. Because we have this kinked tail here, this is going to essentially prevent our interactions. Our interactions are going to be a lot weaker between two molecules if we have this kinked tail, because there's not going to be as much space that these two molecules can interact with one another. So saturated and unsaturated fatty acids are two different types of fatty acid tails, but in terms of the monomers of our lipids, we can see those over here. So our monomers of lipids are composed of a fatty acid molecule and a glycerol backbone. So this here in blue is the glycerol backbone, and this here in gray is showing us the fatty acid tail. So when a glycerol monomer comes together and meets with a fatty acid monomer, we're going to get the polymer, which is known as a lipid. Now I like to look at this here. This makes a lot of sense here because you can see that this sort of looks like an L, and an L that's been this L has been knocked over. But as we look at this here, I'll draw this little L here. This is how we can remember that this is a lipid. These are the monomers of the lipid. You can see that L has been sort of knocked over on its side, but that's one way that I always used to remember that our lipids are composed of these two monomers. One of the other things that you can see on this slide here is the dehydration synthesis reaction. So we're going to have these two hydrogens and an oxygen that are going to sort of meet at this location on our molecules. And when the dehydration synthesis reaction is going to be taking place, we're going to be removing water and we're going to be creating this bond, and that's going to turn this into a lipid. Now, phospholipids, as I mentioned, are very, very special types of lipids, and they are going to be our major component that can be found in our cell's membrane. So the cell membrane of all of our cells are going to be made up of these different phospholipids, and there's some other things that are found in our membrane, but phospholipids are the major component of our different cell membranes. What is so unique about phospholipids are these molecules are what are known as amphipathic. Amphipathic molecules are unique because they are going to have a nonpolar part of the molecule and a polar part of the molecule, all in the same connection of constituents. So you can see that this molecule is going to have a polar head and nonpolar tails. Remember, nonpolar molecules are going to have lots of different hydrogens, and they're going to have equal charges throughout their entire structure. While polar molecules are going to have an unequal distribution and sharing of charges throughout the molecule. So this head up here of our phospholipid is going to be polar, and these tails are going to be nonpolar. Now, on top of this being a little confusing, why on earth, why does this matter? Why does it matter that we have an amphipathic structure with our phospholipids? Well, it all comes down to the interactions that are able to take place through our phospholipids that give us the cell membrane. So polar molecules are going to be attracted to other polar molecules, and nonpolar molecules are going to be attracted to nonpolar molecules. So if we have a whole bunch of phospholipids that all have polar heads and nonpolar tails, one thing that we can see taking place here is that the polar heads are all going to be attracted to each other, and that's going to form sort of a barrier. And those nonpolar tails are going to be also connecting with each other. And then with another layer of those phospholipids, those are going to sort of attach down here, and then these different polar heads are going to be found out here. So the cell membrane is formed in this way. So we can actually see that this is showing us what the cell membrane looks like. So at the microscopic level, even smaller than the microscopic level, at the chemical level, it's important for us to understand the way that these phospholipids are going to interact interact with one another to form the cell's membrane. So what we do, what we call this cell membrane here, is we have two different layers of phospholipids. We have this, we could say that this will be the outer layer, and this is going to be the inner layer. So what we say, in terms of the cell membrane, we say that we have a phospholipid bilayer, and that's how we refer to our cell's plasma membrane. Now, there are going to be a lot of other things that can be found in our cell's membrane as well, such as proteins. But that being said, these are going to be the major component. These sort of form the base of our cell's membrane, and it's all due to the amphipathic structure of phospholipids. So this is important. This is written up here in red. I definitely want you to know what amphipathic means. It means that a molecule is going to have one polar part and one nonpolar part. And as I also mentioned earlier, we will touch more on phospholipids in Chapter 3. So our next type of organic compound are proteins. Proteins are going to be providing many different functions to the body, and you can see some of these functions here. So they're going to give a structure to our body. They're going to give shape. They're going to help to regulate different processes and chemical reactions throughout the body. They're going to provide different types of protection. There's lots of protective functions of proteins. Muscle contraction is going to be facilitated through different types of proteins. Different substances will be transported through the body by proteins, and proteins will also function as enzymes. So there are quite a few functions of proteins, so it's important to know the structure of our proteins so that we can understand these different functions. So you can see these are the types of proteins we see on the left here, and their functions are going to be listed on the right. So our structural proteins will be giving structure to the body. So that's going to be the function of our structural proteins. They're going to be providing a framework for different parts of our bodies. So one good example of this is keratin. Keratin is a type of protein that can be found in fingernails, and they make the rigid structure of fingernails. They make the fingernails very, very hard. That's a lot different from a lot of our other body structures. So certain proteins can provide a structural function to give shape and structure to different parts of the body. So regulatory proteins are going to be functioning as hormones. Hormones, if we can remember from Chapter 1's lecture video, they are going to be regulating different body processes. They're going to be different chemical messengers that are going to help to control the body's growth and development. So there's a lot of different function to regulatory proteins, but these are just going to be functioning as hormones. One example that we have of a regulatory protein is insulin. Insulin is going to be a very, very important protein we have in our body that's going to be helping to regulate our blood glucose levels. So the pancreas is going to be releasing insulin when we eat sugar, and insulin is going to actually be functioning to help to reduce blood sugar. So insulin is a very, very important regulatory protein. We have contractile proteins. The contractile proteins are, of course, going to function to contract. So they are going to be the proteins that cause our muscle cells to shorten in length and to contract and to produce force. And in turn, this is going to allow our bodies to move. So contractile proteins are going to be a major component of our muscles. Two of the contractile proteins that we will see and we'll learn about in Chapter 10 are myosin and actin. So myosin and actin are two different types of contractile proteins that are going to work together in order to contract muscles. Immunological proteins are going to be proteins that are going to have some type of immune system function. So for example, an antibody. Antibodies are going to be different proteins that are going to be very specific to what they attach onto. So antibodies are sort of shaped like a little Y. So I've just taken the liberty of drawing this little Y shape here. This is roughly what an antibody is going to look like. And so there's two different regions. There's this region down here, and then there's this region up here. And so antibodies are going to be specific, and they're going to bind to particular different pathogens that have entered our bodies, and they're going to serve a protective function. So this end is specific, and it might attach to some type of bacterial cell, while this end is going to signal to our own body cells that they're going to tell our cells, like, hey cells, I have a bacteria, and you need to attach to me and take this bacteria and destroy it. So antibodies are going to be a type of immunological protein, and those are going to help defend our bodies against foreign substances and invading pathogens. Now, pathogens are any different type of bacteria or a virus or protozoan or anything of the sort that are going to serve as a dangerous invader to the body. So pathogens are any things that can cause infections, and we don't like these. We don't want to have these, and so antibodies are very good to aid in keeping these at bay. Certain proteins can be classified as transport proteins, such as hemoglobin. Hemoglobin is going to be an important protein that's found in our red blood cells. And hemoglobin is going to be attaching to oxygen and helping to carry it throughout the body in our red blood cells. So proteins can have a transport function as well. They can transport several things throughout the bodies. And lastly are the catalytic proteins, which function as enzymes. And this is going to be one of the types of proteins we talk about the most frequently. So enzymes are going to be functioning as catalysts for reactions. They're going to speed up reaction speed, and they will regulate different reactions that occur within the body. So salivary amylase is a good example. This is an enzyme that is found within saliva. And this enzyme is going to start breaking down food before you actually even swallow food. So we'll learn more about salivary amylase as we go. But ATPase is going to break down a molecule known as ATP, which I will be talking about at the end of this presentation. So there's many different enzymes that can be found within the body. But one thing that I'd like to mention about enzymes is that whenever we have an enzyme, we always finish the enzyme off with ASE. So this suffix, ASE or ACE, is going to indicate that we have some type of enzyme. So salivary amylase is an enzyme in the saliva. Sucrase is going to be breaking down sucrose. So sucrase is an enzyme that breaks down sucrose. So this suffix here is going to help us to understand that's when something is an enzyme. So the enzyme's name is going to tell you that it in fact is an enzyme. That's going to bring us into our monomers of proteins, which are known as amino acids. Now there are 20 amino acids. There are 20 common amino acids that our bodies are going to use. Now you guys don't need to know every single amino acid, but for the sake of this slide and understanding this concept, I'm going to tell you that glycine is one amino acid, and alanine is another amino acid. So each of these are going to be different types of amino acids. Now, proteins are going to be long chains of amino acids. So you can get the idea here that the monomer of proteins are amino acids. And with each of these organic compounds, the monomers are going to come together to give us our polymer. So the polymer is the protein itself. So when two amino acids come together, they're going to form what is known as a peptide bond. So this word here, peptide, is referring to protein. So if you ever hear this word peptide, you know that we're talking about some type of protein. So I'm going to tell you that our polymer of proteins is what's known as a polypeptide. And through the same reactions that will link our lipids and our carbohydrates, we have the dehydration synthesis reaction that's going to remove water and create a peptide bond here. So a peptide bond is going to be between two amino acids, and that's going to also give us water. So it's the same reaction that we saw in all of our other types, and this reaction is going to function in the same way, but it can be used for proteins, carbohydrates, and lipids as well. So as you might guess, if we have two different amino acids, we have a glycosylalanine now, that's a big name there, but this is now going to be called a dipeptide because we have two amino acids that were linked, and that's going to give us a dipeptide. Now, one of the most important concepts that we are going to study about proteins are the individual structural organizations of proteins themselves. So, I've constantly been saying since we've started this class that structure is correlated with function, and this is no different here with proteins. Proteins especially are going to receive their functions from their structures, and that's why we have a structural organization of our proteins. So you can see that we have a primary structure, we have a secondary structure, a tertiary structure, and lastly a quaternary structure. So proteins have these four different structures, and this is the way that we can break up these individual parts of proteins and see how they're going to interact with one another. So first and foremost is the primary structure. The primary structure of our proteins refers to the exact amino acid sequence that is making up our polypeptide. So we can see that this is going to be one amino acid, and that's going to be linked to another, and then we have these peptide bonds in here and so forth, and we're going to have this chain of amino acids. And regardless of what, we mentioned glycine and alanine, so I'll just use these as an example. Let's say we have a glycine here, and we have a glycine here, and then we have an alanine, and then we have another glycine. That's going to be referring to our primary structure. So the sequence of amino acids. So the primary structure is glycine, glycine, alanine, glycine, based on what I just said there. So again, these amino acids are not specifically glycine or alanine. These are just sort of non-specific amino acids so that we can understand the picture. But that's how we would look at this. If this was a glycine, and this was a glycine, and this was an alanine, and this was a glycine, our primary structure would be the sequence. So polypeptide chains can be very, very long. And if we have the sequence of amino acids, that's going to be our primary structure. The secondary structure is going to be different versions of how this primary structure is going to fold on itself. So what proteins are is they are just long chains of amino acids that have folded in particular shapes. And that shape is going to give us what we know as our protein's function. So with this picture here, we can see that there are many different types of amino acids shown. We can see that here's one, and then we have here, and then there's going to be some other ones here, and so forth. And there's going to be a long chain of amino acids. They're all going to be linked up together. But one thing that we can see is based on the specific amino acids, that's going to potentially allow for the formation of hydrogen bonds. So remember hydrogen bonds, and from our last part 1 of chemistry. With hydrogen bonding, we have a partial negative charge meeting up with a partial positive charge. So let's say that we were to, maybe we have a partial positive charge here, and we have a partial negative charge down here. And so as this protein is going to be created, this protein is going to start folding upon itself. And then those two are going to be, as we wrap around here, this amino acid is going to have an oxygen that happens to stick off here. This amino acid is going to have a hydrogen that happens to stick off here, and that is going to form a hydrogen bond to keep the shape intact for this protein structure. So the secondary structure is formed by these individual interactions, specifically our hydrogen bonds, which is another reason our hydrogen bonds are so important. So there's two types of secondary structures within our proteins. We have the alpha helix and we have the beta sheet. The alpha helix you can see is named because it makes a helical structure as it spirals in any particular direction. And the beta sheet makes a sheet. So you can see that this sort of creates different structures which are going to provide different strength in certain areas. They're going to provide different binding sites and so forth, depending on the type of protein we're looking at. So you can see in this picture we have this spiral portion here. This is an alpha helix. And you can see that this is an alpha helix over here. And so we might have, maybe we have a really, really long chain of amino acids, and there's going to be an alpha helix at the beginning, and maybe there's going to be an alpha helix at the end too, or maybe there's going to be 3, maybe there's going to be 1 in between. You can see that there's a beta sheet that is shown right here. So this is going to be indicating us a beta sheet. So this is one way that our proteins can fold. So beta sheets and alpha helices are going to be our two different types of secondary structures. Our secondary structures are going to give our proteins very, very special properties. And they will also refer to only one part of our whole peptide chain, our polypeptide chain. So you can see again in this picture, we have what looks like a long ribbon here. This is a long ribbon, and it's just going along, and we can follow this with our eyes and so forth. So these are all different amino acids that are creating this big polypeptide. So this is a polypeptide chain, and you can see that certain parts are turned into some of these secondary structures. Now, the entire polypeptide chain that we're looking at is going to give us our tertiary structure. So overall, how this entire tertiary structure is going to end up in the overall shape of our entire polypeptide chain, that's going to refer to our tertiary structure. So the overall three-dimensional shape of our polypeptide chain is the tertiary structure. So I always used to get the secondary and the tertiary structure confused. The tertiary structure refers to an entire polypeptide chain and how it is going to look three-dimensionally, while the secondary structure refers to only a part of that polypeptide chain and how that part is going to fold three-dimensionally. Lastly is the quaternary structure, and the quaternary structure refers to multiple polypeptide chains that have linked together to give us what we know as a protein. So you can see that we have our gray polypeptide chain and we have our blue polypeptide chain, and those have now come together to give us two different subunits, and now that we have two subunits, that's going to give us our protein. So the quaternary structure refers to all of the overall protein shape, essentially all of the polypeptide chains and how they are combined to form our overall protein. So that's going to take us into enzymes. Enzymes are very, very special molecules that can be found throughout the body. They serve as catalysts, as I mentioned in the last class. So again, catalysts are going to be functioning for increasing the reaction speed. Enzymes are what are called highly specific. They are very, very specific, which means that they're going to have a very, very unique binding site that's going to allow our enzymes to bind to a very few amount of molecules, or perhaps even just one molecule. Only one molecule will be able to fit into an enzyme's binding site. So enzymes are extremely important for our body's function. Without enzymes, the chemical reactions in our bodies, some of them would move too slowly for us to sustain life. So enzymes increase efficiency incredibly. They are they are amazing in how quickly they can speed up chemical reactions. Some enzymes can speed up reactions 100,000 times. So these are very, very efficient. We see on this slide the example of an enzyme. So this large orange thing here is going to be representing our enzyme. And every enzyme is going to have what is known as an active site. So the active site is going to be the very specific site that's going to be accepting some other type of molecule. And the molecule that binds at the active site of our enzyme is what is called a substrate. So these are more generalized terms that we can use to understand enzymes as a whole, but each enzyme is going to have its own unique substrate. So every enzyme has a substrate, and those substrates might be different. So this enzyme is showing us sucrase. Remember, sucrase is going to be the enzyme that breaks down sucrose. The suffix here, ase, is going to tell us that we're looking at an enzyme. So sucrase refers to this enzyme here. It's going to have an active site that's going to allow for sucrose and water to come in and bind, and that's going to catalyze this reaction. So the substrate's going to meet at the active site of our enzyme, and then the enzyme is going to catalyze this reaction, speed it up, and in this particular reaction, you can see, is going to break down sucrose into its substituent parts. So you can remember sucrose was glucose and fructose together. These different constituents are then going to pop out, and this enzyme is going to become available for other sucrose molecules to come in and be hydrolyzed. So this picture is nice. It shows this very, very simplified version of how an enzyme is going to take its substrate, break it down, and then it's going to spit those parts out, and then it's going to be available for new substituents. So this is the overall view of an enzyme. Our last type of organic compound are nucleic acids. Our two major types of nucleic acids are DNA and RNA. Both of these molecules are going to have their own unique functions. DNA is going to be forming the genetic code for our different organisms. So every person is going to have their own unique DNA. So each of you listening to this class is going to have your own unique DNA code, and that's going to be made up of nucleic acid. And we also have RNA that's going to help with the formation of different proteins, which we will discuss in Chapter 3. So just like our carbohydrates, lipids, and proteins, we also have a monomer form of nucleic acids. And the monomer of our nucleic acid is known as a nucleotide. So right here you can see that this is showing us a nucleotide. So let's unpack this slide. There's quite a few different options in either of these boxes. Let's talk about what makes up a nucleotide. So a nucleotide is going to be composed of three different things. Each nucleotide is going to be made up of a phosphate group, shown here in blue, a pentose sugar, shown here in purple, and a nitrogenous base, shown here in yellow. So these are the three components of our nucleotides, and nucleotides are the monomers of our nucleic acids. So every nucleotide is going to have a phosphate group, and the phosphate group's always going to be the same. So this one's pretty straightforward. But depending on the type of nucleic acid that we have, and which particular nucleotide we're talking about, that's going to be indicating these other two groups here. So whichever the pentose sugar is, ribose or deoxyribose, if you remember back on the carbohydrate slide when I showed you the different monomers of carbohydrates, or the monosaccharides, these deoxyribose and ribose were two different types of pentose sugars. So I'll let you reference that on your own, but that is there for you if you'd like to go back and double check that these are the same molecules. But our pentose sugars are going to be, it's going to be one of these two types of pentose sugars that's going to be found making up this pentose sugar group. So in DNA, we'll have deoxyribose, and in RNA, we're going to have ribose. And actually, DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. So that's a mouthful, but that's actually named. DNA is named because it has this deoxyribose sugar making up its backbone. Ribose, on the other hand, is going to be called making up ribonucleic acid, or RNA. So ribose or deoxyribose, whichever pentose sugar we're having in our nucleotide, that's going to determine whether we're looking at RNA or DNA. The nitrogenous bases, on the other hand, there's going to be a few different groupings of these. So we have the pyrimidines and we have the purines. The pyrimidines are cytosine, thymine, and uracil, and the purines are adenine and guanine. So depending again on which type of nucleic acid we're looking at, whether it's DNA or RNA, that's going to determine which different nitrogenous bases we can put in this group here. So the nitrogenous base is very unique because this is going to be where we hold the code for our DNA. So the phosphate group and the pentose sugar make up the backbone of our nucleic acid, and the nitrogenous base is going to house the code. So one of the most important concepts of DNA here that I'm going to mention is that there's a very unique structure of DNA, and that is known as a double helix. The structure of DNA is a double helix, which is sort of this spiraled ladder. It sort of looks like a ladder that's been twisted here. And that is what is known as a double helix. Now, one thing that I'd like to mention about this double helix is you can actually see the phosphate groups and the different deoxyribose sugars here making up the backbone on our nucleic acid here. This is DNA specifically, so we're looking at deoxyribose sugar. And then those nitrogenous bases are going to be found in the center, and they're going to be connecting to each other, forming different hydrogen bonds across the center of our nucleic acid. So before I go any further into the structure, I want to note that uracil is only going to be found in RNA, and thymine is only going to be found in DNA. The rest of these different types of nitrogenous bases can be found in both RNA and DNA. But if we are considering DNA, we will only have thymine, and if we're considering RNA, we will only have uracil. Uracil will never exist in DNA. So if you see uracil, you automatically know that we're talking about RNA. So this here is a phosphate group in blue, and we have our deoxyribose sugar here shown in purple. So if this is deoxyribose, we're going to be referring to DNA. Therefore, we will not see any uracil nitrogenous bases in this picture. But you can see up here that we have these phosphate groups and the deoxyribose sugar that are linking to each other, and that's what's forming the backbone of our DNA, sort of the ends of our ladder, or the legs of our ladder on either side. And then these different nitrogenous base pairings in the center are going to refer to our different code that we have. So one thing that's important to mention with DNA is that adenine is always going to be hydrogen bound in the center with thymine. Adenine and thymine always bind together. So you'll always have an arrangement of A binding to T. And if we have T on one strand of DNA, we're going to always bind that with A. So A and T are best friends, and they will always be together. They will always bind together unless we're looking at RNA. But in DNA, A and T will always bind together. And therefore, cytosine and guanine will always bind together in our DNA as well as in our RNA. So cytosine and guanine will always bind together. So we always will have C bound to G, or we will also always have G that is bound to C. So this is always going to be true in both DNA and RNA. This is going to be true only in DNA, and this is only going to be different because in RNA we'll have uracil, and uracil will be binding with adenine in thiamine's place. So each one of these nucleotides, this nucleotides being the name for the phosphate deoxyribose sugar here in the nitrogenous base, so that's going to be our nucleotide. These nucleotides are going to be stacking upon one another, and that's why we make, these are our monomers of our nucleic acid. So a DNA molecule is just a long strand of nucleotides that are bound together by the backbones and by hydrogen bonds in the center. So you can see that A here is always going to be bound to T, and T is always going to be bound to A, while G is always going to be bound to C and vice versa. So you can see our different nitrogenous bases shown down here. You can see that adenine will always pair with thymine, and guanine will always pair with cytosine in DNA. In RNA, adenine will always bind to uracil. Now you can see the arrangements of these hydrogen bonds here in the center, which are going to keep our double-stranded, that's why we have a double helix. You can see these hydrogen bonds here are formed between this hydrogen and this oxygen, and this nitrogen and this hydrogen in our thymine and adenine. With cytosine, we actually will form 3, and this will be actually considered a stronger bond because there are 3 hydrogen bonds as compared to up here where we have 2. So again, when we look at the components of our nucleotides, that's going to tell us all we need to know about our DNA. We'll always have a phosphate group and some type of pentose sugar. If we're in RNA, we're going to have ribose, and if we're in DNA, we're going to have deoxyribose. And the nitrogenous base is going to determine the different code that's going to allow us to form different proteins and so forth. So again, adenine in DNA will always bind to thymine, and thymine will always bind to adenine. That is in DNA only. However, in RNA, adenine will always bind to uracil, and uracil will always bind to adenine. So in RNA here, we will always have A binding to U, and in DNA we'll have A binding to T. Cytosine and guanine will always bind together no matter the different type of nucleic acid we're talking about. So C will always bind to G. So if I was to show you a depiction that included uracil, you could already tell me that we have a bunch of ribose sugars because uracil can only be found in RNA. So on this picture, this is going to be a chart that allows us to compare and contrast DNA and RNA. So this is be hopefully helpful for you all. So the nitrogenous bases that are found in DNA, you can see that adenine, cytosine, and guanine are all going to be found in both RNA and DNA. However, in DNA, we have thymine, and in RNA, we have uracil. The different type of pentose sugar is going to be, in DNA, is going to be deoxyribose, and we'll have ribose found in RNA. Now DNA, as I mentioned, is going to form those different base pairs. We're going to have those hydrogen bonds forming our double helix. You can see the double helical structure here. And so this is always going to be the way our DNA is going to look. It's always going to have 2 different strands. Now RNA is only going to have one of these strands, so it's only going to be mimicking one side of this. That's going to be our RNA. You can see that A will always bind to T in DNA with 2 hydrogen bonds, and A will bind to uracil in RNA with 2 hydrogen bonds. Guanine and cytosine will always bind to each other with 3 hydrogen bonds in both DNA and RNA. One of the things that we're going to learn in Chapter 3 is that DNA self-replicates itself, while RNA is actually going to use DNA as a blueprint. So for right now, don't worry about this until we get to Chapter 3, but we will learn about protein formation and how RNA is actually going to be using DNA as a blueprint. And lastly here, DNA is going to carry the code for different proteins. So the specific proteins that you have within your body is going to be encoded on the strand of DNA, and this is going to be based on the sequence of nucleotides. So is it an A, is it a G and then a C, or is it an A, a T, and then a C? You know, it just depends on your unique code. But RNA is going to be helping to create proteins, while DNA is going to be carrying the information that RNA uses to make those proteins. So this is a compare and contrasting view here of RNA versus DNA. Now lastly, for our organic compounds chapter here, in chapter 2, we're going to be completely finished with chapter 2 here in just a minute. But there's a very special nucleotide that I'm going to mention, and that is known as adenosine triphosphate. So you can see an adenine, a nitrogenous base, is going to be linked to a ribose sugar. So adenine and ribose make what is known as adenosine, and that's where we get this name here. So when there's an adenine and a ribose, we have adenosine. And this particular nucleotide is going to have 3 different phosphate groups. So we call this ATP. ATP is our primary energy storing molecule in the body. This is our body's energy currency. This is what fuel our body is going to run on. And so whenever our body's creating energy, we need to create ATP from other methods. So we will be touching on ATP quite a bit. We're going to be talking about this molecule for the rest of the semester, but this is in fact a very special nucleotide, which is going to have 3 different phosphates. This is the major difference here. Instead of having 1 phosphate, we have 3 phosphates. And because we have 3 phosphates, you can see that there's going to be energy stored in these chemical bonds here. And so ATP, after undergoing a hydrolysis reaction, hydrolysis, so the breaking of bonds, when we break this bond here, that's going to release chemical energy and allow our bodies to use that energy for our own body's regulatory purposes. So that will conclude our chapter 2 video here on chemistry. Again, we're going to end up here with ATP. This is going to be a big concept for the rest of the semester, so make sure you're comfortable on this and be ready to learn a lot more about this in chapter 3, and especially chapter 25, which we will cover right after chapter 3. So if there are any questions about the material presented in chapter 2, please feel free to send me a message, but otherwise I will see you all in the next video.